Welcome back to our channels. Previously, we talked about membrane, okay? Structure of the membrane and some membrane transport basic stuff. And now we are on the HL part, more complicated about the details of cell membrane and also about why we have the model, the evolution of the model of the cell membrane, the plasma membrane. So welcome to our biology channel again. We are on 2.1, okay, second part, okay, and membranes and membrane transport too. And this is completely additional higher level for this one, so more complicated. And we are still on the form and function in cells, okay? And we talk about 2.1, about the membrane and the membrane transport, okay? First, we have the fluid mosaic models, okay, about the cell membrane. What's the purpose of the cell membrane? And by this model, the phospholipid bilayer of the cell membrane acts as a two-dimensional fluid, okay? Yeah, it is a fluid, okay? And this kind of fluid, it provides the fluidity and permeability of the cell membrane, okay? Allowing the movement of protein on it, along it, or fluid, okay? And this is the model, 2D structure of the fluid mosaic model. It is an evolution from another model. We'll talk about it in details later. And when you join the fluid mosaic model, you need to draw at least one kind of protein there, okay? Every kind of protein we have to draw one, okay? For example, peripheral protein on the surface and a glycoprotein for cell-cell recognition with a carbohydrate chain, okay? And the cholesterol for stabilizing the bilayer, giving it fluidity or yeah, and then for the channel protein, the going fluid, okay, is a pro here for iron to going fluid, okay, one side or two side or to co transport, okay, and there are some different kinds of protein besides this one. One is the integral protein inside the bilayer but not going fluid, and the one called transmembrane protein is exactly going fluid, okay. So remember, I've said that in IBDP requirement, in join the fluid mosaic model to give you a notch of marks. So these are one integral protein, one protein channel, and one peripheral protein, glycoprotein, and one cholesterol have to be drawn, okay, separate, okay, and you need to be labeling. And actually, this model, mosaic model, is an evolution from the old model. Then the old model has ruled the world for 40 years. Okay, it's a Danieli model. At the 1935, okay, it proposed this model. It said that the cell membrane has three layers. Okay, one layer of protein, one layer of bilayer, phospholipids, and then the other protein. Okay, three layers. One layer of protein outside, and one layer of protein inside the cell. Okay. But why they have this model? Because they have some observation. Okay, they said the plasma membrane is partially permeable with organic solvent. Okay, passing through it faster than water, so it has to have a hydrophobic layer inside, right? And the new model agree with it too. And the other thing is they have the chemical analysis showing that the plasma membrane consists mainly of lipid and protein. So where is the lipid protein? Okay, yeah, and then they observe it by the early electron microscope. They look at the plasma membrane. They can very distinguish as three bands, okay, three layers. So one is a light band and two dark bands. So definitely think that, okay, the light band is the lipid layer, okay, bilayer, okay, and the two dark band is the protein, okay. It is a very preliminary one, but because of the low resolution of the electron microscope at that time, okay, it is the best guess, okay. And it rules it for 40 years until single Nicholson model appears because they have a better resolution of the new electronic microscope. By single Nicholson model, cell membrane only has one phospholipid layers by layers. The other proteins have different flows on in the phospholipids bilayer fluid. Okay, they flow on a different way to flow on a peripheral cholesterol to support it. Okay, channel one with pores, going through it integral. Okay, or transmembrane. They have different names. Okay, there are different types of membrane protein. Hydrophilic protein on the surface. Okay, hydrophobic protein inside the hydrophobic region. Okay, peripheral protein penetrating to the hydrophobic region, and integral protein passing through the membrane. they have different names. Okay. It was proposed in 1972 after the Davidson Danieli model worked for more than 40 years. But why it appears suddenly? Because of high-resolution electron microscope, they observed the hydrophobic protein observed inside the cell membrane. And they also found out hydrophobic region inside the phospholipid bilayer. And later from fluorescent labeling, even they can have other kind of membrane protein being observed partially or right through the phospholipid bilayer are observed. So the existence of polyphenols and indigo protein are lots of different names. Okay. So dependent of the fluidity, we said that how do the two fatty acid change, okay, being changed, okay. So before you look at here, okay, here you only have the chain here. This should be very irregular, okay, a high fluidity, right, and easy to permeable fluidity, too high, okay. And how can it be regulated to become not so fluid and not so permeable? It is helped by the cholesterol, okay. It's inside the model. Cholesterol is with the enhancement of regularity, okay, by the cholesterol, okay, molecule are harder to pass through the membrane. So the cholesterol also reduce the membrane permeability. Both so the fluidity and the permeability has been reduced because it's too high. Too high. So fatty acid composition of fluidity. Fatty acid composition of lipid bilayer affect is fluidity. Okay. How can it affect it? Depends on the interaction between this fatty acid. Okay, if it has an unsaturated fatty acid, okay, remember more double bond, right? More double bond is less bonding with other, so it has a lower melting point, less interaction between the fatty acid, so lower melting point. So the membrane are more fluid and therefore more flexible at temperature experienced by the cell. Okay, higher temperature. And if it is a saturated fatty acid that has more interaction, is a higher melting point. Okay, so the membrane are stronger at higher temperature. So how can the membrane change? Okay, so in cold habitat, very cold, right? Freeze. Okay, so the fatty acid might freeze with each other, so it has to have a lower melting point. Okay, so that it doesn't freeze. Okay, to keep its fluidity. So in cold habitat, low melting point, so more saturated or more unsaturated? Yes, more unsaturated. Okay, fatty acid are there. Okay, it is an observation. Okay, so it has a more unsaturated fatty acid chain there. Okay, and this unsaturated fatty acid chain to lower the melting point and keep it fluid. Okay, and prevent them closely packing or stiffening together. Okay. So we have the bidirectional regulation of membrane fluidity, and this is the role of the cholesterol again. Before we just say the cholesterol has one direction, but actually two directions. In high temperature, okay, well, too fluid, okay, and the cholesterol make it more regular. In the low temperature, wow, stiffening, can go together, and the cholesterol make them a lot joined together, not bounce together, okay. So cholesterol acts as a modulator of the membrane fluidity, stabilizing the membrane at higher temperature to fluid, okay, and preventing stiffening at lower temperature. Okay. So at high temperature, the lipid in the membrane becomes more energetic and move around okay, too energetic, and the fluidity and permeability is too high. And then low temperature on the other hand, the lipid and the membrane has loose energy and they're rarely packed together, and the fluidity and permeability is too low. Okay, in low temperature. So in high temperature, cholesterol stabilize the membrane. Don't let them move too much. Okay, reducing the fluidity and permeability. At low temperature, the cholesterol separated the phospholipid, the fatty acid, okay, preventing them from clustering together, enhancing the fluidity and permeability. So cholesterol has a bidirectional regularity of membrane fluidity. You have to remember how to explain it in the low temperature and high temperature. In low temperature, more unsaturated fatty acid. Moreover, cholesterol healthy too. Okay. And then we talk about how transport. Okay, how the membrane okay help to transport material into the cell and out of the cell. Into the cell, we call it endocytosis. Okay. Moving material out of the cell, we call it exocytosis. Okay. And all are through vesicle. Okay. Vesicle is a single membrane surrounding molecule inside a cell, and this single membrane can made up of phospholipid bilayer. So we trust that it come from the cell membrane also, and also come from the membrane of the organelle. Okay. And it can move around. Endocytosis is the taking in the molecules by a living cell. Okay, so at the surface of the cell membrane, okay, it engulfed the materials and forming a vesicle. Means that part of its cell membrane is broken into vesicle, and then the molecule in the vesicle can move around inside the cell until it go to the organelles that it target at. Okay, and then the vesicle will be fused with the cell membrane of the organelle, or the membrane of the plasma membrane of the organelle, and then fuse together and then burst, and then the material can go inside. Okay, we call it endocytosis. And then exocytosis is the opposite. The organelle want to release some material. They make some material, want to release it. Okay, so the plasma membrane of the organelles, okay, becomes a vesicle, and the vesicle move around, and then the vesicle fuse with the cell membrane because there's the same structure, bilayer structure. Okay, and then bursts out. Okay, what's the example? Yeah, it's a transport in the vesicle. Okay, so material moving within the cell. Okay? So metabolism of organelles needs input molecule and it also produce a molecule. So you need to get the material in and then get the material out. Okay, and then this molecule will move within the cell in vesicle. Okay, it is a single membrane surrounding the molecules. A vesicle can fuse with the membrane of other organelle. Okay, just it can transport molecules to other organelle. A vacuole, okay, is larger than a vesicle, okay, but a vacuole cannot fuse with the membrane of other organelle. That's just a vacuole cannot transport molecule to other organelle. Remember, single layer and double layer member. For example, the rougher endoplasmic reticulum may produces bursts of proteins, okay, and this protein bursts off, okay, by its plasma membrane, okay, and become a vesicle, and this vesicle, this protein is not mature. It is to go into the Golgi apparatus, fuse together, and the Golgi apparatus makes the protein mature for other usage outside, and then it becomes also mature protein is released as vesicle again, and the vesicle go to the cell membrane and burst out. Okay. Exocytosis. This is the process example. Okay. Of course, there's some organelle vesicle by themselves. For example, lysosome. Okay. And then we talk about the gate ion channels in neuron. Remember, inside the cell membrane there is a channel. There is a gate ion channel for there's a port, okay, for ion to pass through, specific ion to pass through. We call it gated ion channels. Okay, it's a protein for ions to pass through the cell membrane. You mean in and out of the cell. Okay. Oh, for the ion channels, for example, in the neurons, okay, it's a gated port, and then in this gated port, opening and closing is regulated by two factors. Okay, for example, by some material called neurotransmitter or by the voltage. Okay. For example, the neurotransmitter gated ion channel is the nicotinol acetylcholine receptor. Okay, the receptor transduce chemical signal, the chemicals, into an electrical signal in the neuron. Okay, and also in the neuron we have some voltage gated channel. Okay, we call it sodium potassium channels. Okay, in signal transmitter, sodium enters the neuron and then potassium exits the neuron. Okay, in normal signal transmission, so we have to move them back, right? So the sodium potassium pump is to use it to regulate or restore the gradient of sodium and potassium ions. Okay, after the transmission of this signal. Okay, and we call two kinds of transporter. Okay, two kinds. Okay, one is an exchange transporter. Okay, we really transport that way in a specific direction. For example, the sodium potassium pump. Okay, a transporter is integral the membrane of protein. Okay, and it is a pore. Okay, and so in the plasma membrane. Okay, It allows passage of various type of molecule fluid, okay, for example, iron, small molecule, and protein fluid, okay. An exchange transporter is a special one, okay. It allows a one molecule to travel one direction followed by the passage of another type of molecule in the opposite direction. So it is a smart way, okay, we'll learn it later, okay. So it's a channel, it pumps the sodium to one side, and then when it opens the other side, it pumps the potassium to the other side. So it's an efficient way, okay. One channel, two finger transport, okay, both in one direction. So sodium potassium pump, okay, because it is it's an M side, we call it ATP ATPase, okay, ATPase, okay. It just carry a protein of active transport of both sodium and potassium. It is an example of exchange transport. You will encounter it very soon, okay. You have to generate, regulate, and restore the gradient of sodium potassium ion across a membrane. Okay, in exchange transport, the specific ion is being transferred by themselves, okay. And the other, we call it indirect active transport. It is a co-transporter. Okay. The sodium depends on the glucose transporter. So when the sodium move through this transporter, the glucose move together with it. Okay. Indirect active transporter use the downhill flow of an ion. Okay, to pump some other molecules of ion against the gradient. So on one part of it, there's a higher sodium concentration, so sodium go through the transporter. But at that time, actually, the glucose is a low concentration, but moving through by the sodium, okay, sodium flux the glucose out, okay, up the gradient, against the gradient. So sodium dependence glucose transport is an example of indirect active transport, okay. The glucose coupling, okay, with the sodium, they pass through the contrast co-transported together against the glucose gradient. For example, in the small intestine, okay, you need to absorb the glucose, okay, absorb as much as possible, even when the outside is the glucose level is low, it still want to absorb more, so it's against the gradient. And later we learn something called CAM, okay, cell adhesion molecule. We just learn it a little bit, not in details, because IBDP doesn't want you to go too deep on it. You just need to remember, okay, CAM is some kind of molecules for what, okay, is for the interaction between a cell, okay. There are four families of CAMs, okay. Adhesion molecules are cell surface protein, okay. It's a protein also that mediate the interaction between cells, okay, or between cell and the extracellular object. There are four families of CAMs, okay. Immunoglobulin, like adhesion molecule, integrin, catecholamine, and then selectin. You just need to remember the names, okay. The details is our syllabus, okay. Different families of CAMs are used for different type of cell junction. For example, the coherent form adhesion bonds between cells of the same kind, addicting them to stick together to form the tissue. Tissue is formed, okay, when cells adhere to each other by what? By this coherent, okay. It's our syllabus, okay. You don't need to remember detail, but they will describe it in the question, okay, pass it with the paper, exam paper, you to recognize, okay, by the description, I remember it's CAMs, okay. It's cell adhesion molecules, okay. It helps to adhere molecules together, okay. Okay, thank you for going through with me, okay. This is wonderful discussion about transport through the cell membrane, and we will use them later, okay. So we have some idea in your mind or some different kind of transport, the structure, the protein, okay. And then later on, when we learn them in details, you remember it, okay. It's a good preparation, okay. So see you for next week, okay. Bye.